Hello, and thanks very much for the invitation to be part of this workshop. My title today is Beyond Fingerprinting, Optimizing Individual Connectomes for Behavior Prediction over Reliability. So we've known for a few years now that individual functional connectomes, at least to some degree, are stable and unique. And what this looked like in our hands the first time we noticed this result was that if you calculate a whole brain functional connectivity profile from a number of different individuals, scanned while they're both resting as well as performing a few different cognitive tasks over the period of a few days. And then you just ask the simple question, can you match an individual on the basis of their functional connectivity profile across days and brain states? It turns out that you can do this with a pretty remarkable degree of accuracy. So this is telling us that there is a certain degree of uniqueness and reliability in these connectivity profiles such that you can actually pick someone out from a crowd on the basis of these patterns. And this was exciting to us, and there's been a number of other demonstrations both prior to that and since then that corroborate this observation, that really when you account for some of the shared population level group factors that determine sort of general patterns of connectivity, above and beyond that, it really is individuals that account for the vast majority of the variance in functional connectivity. And so again, this was an exciting result, but I think certainly for us and for many others in the field, the value of individual differences is not necessarily in and of themselves for their own sake. I mean, after all, if we want to identify people, there are better ways of doing it than calculating a functional connectivity profile. We could just look at them or look at their DNA or their actual fingerprints. So the utility of these individual differences in functional connectivity is really going to come in if we can show that there's something in these patterns that relates to a meaningful out-of-scanner behavioral metric about people. And the line of work using functional connectivity to predict behavior is sort of proceeding in tandem with the observation that individuals are identifiable and that these patterns are unique. And so, and this is really where a lot of the excitement comes in when we start to think about connectivity-based biomarkers. And our group in our first paper was able to show that we could predict the trait of fluid intelligence based on functional connectivity patterns. And there's been many other demonstrations of behavioral phenotypes that can be predicted on the basis of functional connectivity. So for example, things like sustained attention and ADHD symptoms of autism, as well as things like personality traits and also other cognitive abilities like reading comprehension. So this is taking the next step of sort of proving that identification is not just sort of the point of the individual differences in and of itself, but rather there is meaningful structure in these patterns that actually relates to real world behavioral outputs of the brain. So I think the fact that both of these lines of work were sort of proceeding in tandem and in parallel and influencing one another, as well as the increased focus generally on reliability and reproducibility, which is certainly well placed, has led a lot of people to sort of make this assumption that if we can increase reliability or fingerprinting or test-retest, individual reliability or however you want to operationalize it, if we can increase the stability of individual functional connectivity profiles, this will somehow entail an increase in behavior prediction. And so there's been a lot of work on the side of trying to optimize reliability via better acquisition methods, better analysis techniques, connectivity pipelines, artifact reduction. And the assumption of this pretty large body of work at this point is that an increase in reliability will entail an increase in sensitivity to behavior. And my argument today is that that's not necessarily the case. And by optimizing purely for reliability without an eye to behavior prediction, we might not be getting the sensitivity that we could be out of these functional connectivity profiles. And rather, we should optimize for behavior prediction right from the start. So to support this argument, I'll first show some both theoretical and empirical evidence for this. So on the theoretical side, if we think about, this idea that reliability entails behavior prediction, we can kind of see that that's not necessarily the case. And so one thing that could be the case, and this was something that we got asked a lot, and we also asked ourselves when we started presenting our initial fingerprinting results, was that, well, okay, there's some degree of uniqueness in these connectivity profiles that can be used to identify individuals, but is that actually meaningful? So we could have a situation where functional connectomes are unique, but not actually meaningful. And so they could be akin to something like barcodes, where barcodes uniquely identify the item that they're attached to, but there's nothing in the pattern of thick or thin bars in the barcode that's actually meaningfully related to any feature of the item that it identifies. So that's one possibility. Another possibility on the flip side is that we could have connectomes that are meaningful, but not necessarily unique. And so you could think about a theoretical situation where we have pairs of participants or even groups of participants whose functional connectomes look very similar to the extent that we would actually potentially mistake them for one another in an identification experiment. But then if we were to look at where these individuals fall in some sort of behavioral space, we might see that individuals with very similar connectomes are also very similar on some behavioral metric. And so in this case, the sort of lack of uniqueness might actually help us in the sense that it might reflect meaningful similarities in behavior that could be used to improve our behavior prediction models. Another piece of theoretical evidence as to why improving reliability won't necessarily entail an improvement in behavior prediction across the board is that there's many state level or within subject differences that we might want to be sensitive to. And so one big example of this could be something like attentional states. And by definition, if we're optimizing individual functional connectomes to be maximally stable and unchanging within an individual, we're not going to be sensitive to these sort of within subject state level changes. So one example is attentional states, where we might want to be able to predict if someone is on task or mind wandering. There's many other states that we might be interested in along various timescales. So things like mood or symptom trajectories that kind of wax and wane over the period of days to weeks to months. We might be interested in biomarkers based on or that reflect response to drugs or other interventions. And we might also be interested in longer scale changes like development or aging. And so again, by definition, if we have an individual signature that's extremely stable and unchanging, then we're not going to be sensitive to these state level differences that could be equally, if not more important, to develop biomarkers for than the more trait-like individual differences. There's also an increasing amount of empirical evidence now that suggests that this assumption that maximizing reliability doesn't always entail a corresponding increase in behavior prediction. And this is one piece of work from a few years ago now, where the authors directly showed that the edges or the connections in the functional connectivity matrix over here, the edges that were most reliable or most stable within individuals were not necessarily the ones that were most relevant for behavior prediction. So you can actually see there's not much of a correlation at all. So each dot in this plot is an edge or a connection. And the more reliable edges are not necessarily the ones that are contributing the most to behavior prediction. This is a very recent preprint posted just last week that's showing a similar thing. So in this case, the authors here are showing the edges or the connections that are most important for connectome fingerprinting. So the edges that are most discriminating of individuals here on the left, and the sort of network level connections that are most represented in this set of discriminating edges. And it turns out that there's very little overlap between the edges that are doing the work of fingerprinting and the edges that are doing the work of behavior prediction for these three different behavioral metrics here. So if we were to optimize, again, for just identification or fingerprinting, we might not be optimizing for the meaningful information that allows us to predict behavior. So how do we optimize for behavior prediction? I think that so, this is a basic schematic of what we're trying to do. We're trying to predict behavior as a function of the brain or as a function of some metric that we can derive from neuroimaging. And there's things that we can do on both sides of this equation to try to optimize for this. We'll spend most of the rest of the time talking about things we could do on the brain side, a little bit about behavior. So we could increase our sensitivity when we're actually acquiring the signal, but actually there's things that are even easier and more straightforward that have to do with changing up the brain state that we have people in as we're measuring them. And there's also different things we can do on the analysis side. So beyond functional connectivity, looking at other metrics we can extract from the signal. And so for the remainder of the time, I'll talk mostly about this piece here that we can optimize, and then a little bit on the analysis side. So thinking about brain state, even though a lot of the functional connectivity metrics were developed on resting state, there's increasing amounts of evidence that actually task-based connectivity is much more sensitive to behavioral differences. This is a paper, again, from a couple years ago now, where the authors showed directly that task-based connectivity explains a lot more variance in fluid intelligence than rest connectivity. And they showed this on two different data sets. So on the left here is the Human Connectome Project, a data set of young adults. And on the right is the Penn and neurodevelopmental cohort, which is children and adolescents. And in both cases, they're using data from the same subjects to predict the same target behavior, in this case, fluid intelligence. but they're feeding the model data from different scan conditions, and that's along the x-axis here. And so in both data sets, you can see that the resting state runs actually do the worst in terms of explaining variance in fluid intelligence. And the conditions that do better are the task conditions. So in the case of HCP, gambling, working memory, and emotion, and working memory and emotion echoed over here. So simply by giving the brain something to do, sort of the analogy we like to use is a stress test, by sort of tweaking the circuits in some way, and you are somehow enhancing the sensitivity to these phenotypic differences. One type of task paradigm that I'm particularly excited about is these ideas of naturalistic tasks. So for example, movie watching, this is a really rich, engaging, dynamic thing to have people doing in the scanner. You are involving multiple brain systems kind of simultaneously. And similar to the traditional tasks, we're seeing a boost in prediction accuracy for cognitive abilities as people watch movies compared to when they're simply resting. And so this is, again, same subjects, also from the Human Connectome Project, and the predictions, the distributions in purple come from runs where people are watching movies, and in dark gray, it's resting state runs. And these are all compared to null distributions in light gray here. And so you can see a boost in sensitivity, in some cases a dramatic one for the movie watching as compared to rest. And we also tried using movie watching connectivity to predict socioaffective tendencies. And these are self-report. These tend to be harder to predict overall. So you can see the predictions are overall lower than for cognitive ability, but the only run that was successfully able to predict these tendencies was a movie watching run. And so this is exciting potential for these tasks in helping predict behavior and really draw out and enhance some of those circuits that seem to be relevant for these phenotypes. And I'll show you another piece of data here. So interestingly, these movie runs were comprised, each of them was comprised of a series of shorter clips, so 3 to 4 minute clips. And we noticed an effect such that the clips that had more social content, which was proxied by the number of time points containing at least one face on the screen, was fairly strongly related to the success of the prediction. So in other words, clips that had higher social content were better predictors of cognitive ability. And to kind of drive that home, when we look at the 3 most successful movie clips, they were clips from The Social Network, Ocean's 11, and Inception. So kind of fast-paced, lots of dialogue, engaging clips. And the least successful clips were ones from nature documentaries and just sort of montages without a lot of social information. And so whether this is sort of a general attention or engagement effect, or whether there's something specific to the social content, is something we're still trying to tease out. But I think it really speaks to the importance of choosing the right task, and also even within a movie task, the importance of the content on the screen for really drawing out the sensitivity to these behaviors. A nice thing about task is it's sort of, people that have been in the field for a while, it sort of feels like maybe that we're coming full circle a little bit. But the nice thing about using tasks over rest is that it allows us to get away from connectivity, or to complement functional connectivity with more task evoked activity strategies for data analysis. And so another recent interesting finding, again on the Human Connectome Project data, these authors reported that actually not just functional connectivity, but the task activations, the actual contrast between task conditions, if you build a model on this contrast within individual subjects, you can get a pretty strong prediction of cognitive ability. And in many cases, the task activation contrasts actually outperform the resting state predictions for cognitive ability. And then going back a bit to the naturalistic movie viewing tasks, there's some cool evidence that the so-called typicality of movie watching activity can actually be used to classify a diagnosis of mental illness. So in this case, you're not actually modeling features of the task, but you are looking at the synchrony evoked by the movie in different groups of subjects. And these authors saw that patients with schizophrenia had a lot less synchrony, a lot less stereotyped activity in their movie watching response than healthy controls, so much so that they were able to actually classify on the basis of this typicality whether a subject was a patient or a control. And this approach has been applied also in autism and depression with really interesting results. And I'd like to just hammer home that this is something that can't be done at rest. So you have to have the presence of a ground truth task in order to do these types of analyses. So I've talked a little bit about sort of relatively easy or straightforward things that we can do to optimize things on the brain side of things. I'll just spend a couple seconds talking about optimizing the behavior side. There's a lot of rich work to be done on this side as well. And I think our predictions are only as good as the target behaviors that we're trying to predict. So they're only as valid as the sort of biological validity of the target of our prediction. And there's a lot to be said about this, but I think my sense is that some of the computational phenotyping as well as digital phenotyping efforts will give us phenotypes that, to use an overused phrase, sort of carve nature at its joints a little bit better and get us closer to a biologically valid target of prediction. So to wrap up, today I've argued that individual connectomes do show some degree of stability, but we shouldn't be optimizing necessarily for reliability in and of itself. Rather, we should be optimizing directly for behavior prediction, assuming that that's what we'd like to use these for. And using tasks rather than rest is sort of an easy way to dramatically, in some cases, improve sensitivity to behavior differences. Some of the outstanding challenges in this work, I didn't talk much about making sure that we're getting prediction for the right reasons. And so there's confounds that could be affecting this. For example, head motion can be highly collinear with many of our behaviors of interest. So certainly developing better techniques to handle that will be important. I think we need to balance large N data sets with the need for innovation on task paradigms. So much of this work I showed was done on large publicly available data sets that have either only rest or sort of a rather traditional battery of tasks. And I think that we also need to be innovating on the task paradigm front, which might require smaller data sets. I talked a bit about the last slide about the need for better or more valid behaviors. And finally, I think in this work, longitudinal data sets will really be the gold standard. So actual prediction is what we want to try to achieve. And excitingly, there's some efforts going on that front as well, such as the ABCD study. So with that, I'll wrap up, and thanks for your attention.